everyone. Thank you so much for joining me today for the latest Genome Giants as we take a look at some of the most influential figures in the genomics field. Today, we are joined by Heidi Rehm, who is a geneticist and also a pioneer in implementing genomic medicine. So Heidi, if you could just introduce yourself.
Sure. I'm Heidi Rehm. I sort of consider myself a clinical genomicist, but I wear a few different hats. The Chief Genomics Officer at Mass General Hospital, and I'm an institute member and the medical director of the Broad Institute's clinical laboratory, and a faculty member of the Center for Genomic Medicine at Mass General Hospital.
So we just take it back to kind of the beginning. You kind of spent most of your childhood in like Lake George. Would you just kind of like describe kind of what are some of your fondest memories from growing up?
Yes, so Lake George is in New York, although it's up in a more rural part of New York in the lower Adirondacks. And I grew up on the side of a mountain. My parents owned 40 acres of land. They still live there, actually. And so, you know, we had a pool and lots of space to play outside. And so I think my fondest memories are just playing outside in the woods, you know, in the streams and hiking through the forest and things like that, you know, playing with friends. That's sort of my fondest young memories.
What were you like as a child, and kind of when did you kind of think, oh, like I can kind of see myself getting into like a science career? Because I know that a lot of people kind of start off and they're like interested in something completely different and then they kind of end up in science.
Yeah, you know, in my school was small. There was only 80 students in my whole class, so it was a fairly rural area. You know, I did lots of stuff in school. I was into sports. I played volleyball, field hockey, basketball, softball, so lots of sports. Eventually, focusing more on volleyball and softball. You know, was a good student. I was valedictorian of my class. And so and, you know, had a lot of close friends. So you know, I did a lot of different things in high school. But as much as I, you know, was social with friends and sports and things, I also, you know, was pretty diligent in studying, getting good grades, and graduated valedictorian of my class. And I always had an interest in science. My father was actually a high school biology teacher. And I mean, the interesting thing was I had no desire to be a teacher, in part because the side of teaching that I saw was his frustration with the public school systems. And, you know, coming home from work and being very frustrated with the principal and, you know, policies in the school. And so, you know, I wasn't attracted to wanting to be a teacher at that stage in my life, kind of observing my father and his frustrations from teaching. At the same time, you know, all of the snakes and reptiles and amphibians that he would bring home for the summer that would be at our house and, you know, expose me to a lot of biology. And that might have influenced, I'm sure it did, my, you know, continued interest in science and math as I entered college and so on.
Was that something that you kind of bonded on, and then like as you got older, you carried on kind of bonding over kind of your love of like science and biology?
Yeah, I mean, you know, I always loved the subjects of math and science, and the more it was relatable, the more interesting it was to me. So biology, I felt, was more relatable to the human world around me, so I think that was more of interest. I also am a very type A personality. I like to organize the world, and you know, I remember in probably ninth grade biology, you know, starting to learn about genetics, and to me, like the genetic code was very simple and straightforward and elegant in how, you know, this DNA sequence encoded genes and proteins that, you know, made up what the body does, and that sort of ability to logically understand how the human body works, you know, to me was interesting and fascinating. But also over time, you know, to see how disruption of it would lead to human disease, and wanting to study the impacts of disruption of genes, as I saw that process break down and have effects on people. And so over time, my interest in biology and genetics turned to sort of the clinical impacts of that as I, got further on in my career.
You attended Middlebury College. What kind of made you want to do a BA in molecular biology and biochemistry?
Well, again, that was the sort of field of science and math that was most tangible to me, most relevant to the world around me. So, it was sort of very natural to pick that field. It was the field of biology that most encompassed genetics, which I was, fascinated by. And so, and it was a brand new major. I think I was just the first or second class to be able to take this particular major at Middlebury College, which is a small liberal arts school in Vermont. And I also really enjoyed one of my professors, Bob Kless, who I still am close colleagues with to this day, who was the director of that major. And I did my thesis under him studying Lyme disease, Borrelia burgdorferi.
And then you kind of went on to then like Harvard. Like, what was your experience like here, and how did that kind of compare to your experience at Middlebury College?
Yeah, I remember applying to graduate schools, and at first I didn't have, I applied to Harvard and Dartmouth and Princeton and MIT, and there was a part of me that sort of saw Harvard as this, stuck-up school that I maybe wasn't as down to earth as something that I wanted. And it wasn't actually my first choice because of sort of an impression I had about going to Harvard. But then I went and spent the weekend at a retreat for new incoming accepted students, and just really had a awesome time. I met professors and other students, and everyone was friendly and down to earth and, intellectually stimulating. And it was just an incredible experience, and sort of dispelled my impression of this snooty school. And immediately I was, wanted to attend. And so that was a fairly quick decision to make the decision to go to Harvard Medical School in the PhD program.
And what did you kind of work on during your time at Harvard?
So, I continued to have interest in genetics and particularly human disease. And so when I got to Harvard Medical School, I looked at laboratories that were studying human diseases. Cynthia Morton, Lou Kunkel, Jeff Dewick, these were all human geneticists that studied, different diseases. And, within the 2 rotations that I had done, I quickly decided to stay in Cynthia Morton's lab, and her lab studied hearing loss. And I was fascinated by the inner ear organ, the cochlea. It has this just elegant anatomy where, just understanding the anatomy, you can really understand how hearing happens and works, and how disruption of the different structures can lead to hearing loss. And I got very interested in studying hearing loss, and was actually studying this family with a genetic disease that included hearing loss, but also included blindness and peripheral vascular disease, and a large pedigree in Costa Rica. I ended up ultimately going down to Costa Rica to study this disease in this family.
How did you kind of like, after your time at Harvard, like what did you kind of like, what was your early career kind of like, and how did you kind of transition? And were you kind of like, oh, what do I do now? Because I feel like after I finished my master's, I was kind of like, okay, where do I go from here? And what kind of challenges did you experience at that time as well?
Yeah, so after I finished my PhD, or as I was nearing the end, I continued to be very interested in the genetic basis of hearing loss, and I'd been in a lab that studied the sort of human genetics, molecular biology side of it. And I wanted to stay in the field of hearing loss. I really liked that clinical relevance of understanding the causes of a disease like that. But I wanted to broaden my scope of experience and tools and techniques. So I went to David Corey's lab, who was in the neurobiology department at Harvard Medical School. and he more focused on neurophysiology and understanding physiological basis of hearing. And so the goal was to sort of broaden my base of research skills and tools, but to stay in the field of hearing loss. However, after a couple of years in his lab, an opportunity arose to get involved in a new center that was being created. It was originally called the Harvard Center for Genetics and Genomics. And Raju Kuchilapati had been recruited to start up this new center. And I was asked to start a clinical lab as part of that center. And as I mentioned earlier, I was always interested in the human and the clinical side of science and genetics. And so to me, the opportunity to be involved in a clinical lab that could do genetic testing for patients with diseases and figure out what was the underlying cause of their disease was just really, amazing, to have an opportunity. So I ended up agreeing to take this job and to start to build this new clinical lab as part of this center, the Harvard Partner Center for Genetics and Genomics. And so I was employee number one, and I, started to hire people. Actually, I had to simultaneously enroll in a training program, a 2-year fellowship, to become board certified as a clinical laboratory geneticist, which is the credentials needed to actually be a director of a clinical lab. So I enrolled in that lab while I was starting to build this lab. So I'd rotate at a lab, figure out how they were doing X, and then apply that, as I was building up a team and a lab, and getting the regulatory requirements to be able to actually offer genetic testing. Raju had brought his team from Einstein, who was involved in the Human Genome Project. And so they had scalable approaches to DNA sequencing, or at least scalable at the time. It was still based on the original Sanger sequencing methodology. But we ended up working with that team to apply what was at the time a reasonably high throughput Sanger sequencing platform to genetic testing, and launching some of the first genetic tests for lung cancer treatment, for diagnosing hypertrophic cardiomyopathy, one of the most common monogenic diseases that kills individuals, particularly professional athletes is where you hear about it most. Reggie Lewis, Hank Gathers all died from hypertrophic cardiomyopathy, and we launched the first clinical test for that disorder to be able to diagnose the cause and also identify family members and prevent sudden cardiac death in individuals in those families.
What was it like kind of being approached to kind of launch that, and like, how did you kind of, as you said, you kind of like the first person there, like you were employee number one, how did you like kind of feel during that time, and like kind of what were the challenges around that as well?
Yeah, I mean, it was both fascinating but daunting. I remember taking on this job, and I had to simultaneously, enter this training program. And, I remember, asking, okay, where's the like instruction manual for how to launch a clinical lab, and what are the regulatory requirements that I have to meet? And, where is the book, the guidance, the tools? And basically the answer was there is none. You just sort of figure it out, and you talk to colleagues, and, and I became friends with the clinical labs in the hospitals to understand how they were doing things. Rotating through the labs that I was rotating through as part of my training program allowed me to see the operations and approaches and the regulatory requirements that were happening there. And I just, learned it on the fly. I mean, I remember I had to buy refrigerators and gel rigs and pipettes and freezers. Just there was nothing there. It was all from scratch. And, I hired my first technician when I was pregnant with my first child, who was born 2 weeks after my first technician started. So 5 days after I gave birth, I started coming back to work a half day a week just to meet with her, review what she had done for the past week, and, help guide her for what to do for the next week. And, so I'd, very short maternity leave that first round, I think. And then I came back mostly full time after 5 weeks. So I wouldn't recommend it as, how you should take a maternity leave. My second child, I had a slightly more relaxed maternity leave, but it was hard in the beginning. I had no idea what it was going to take to launch a clinical lab, and it took, over a year to do that.
How did that kind of experience kind of shape the rest of your career? Like the stuff that you learned from that experience kind of drive you like throughout the rest of your career?
Yeah, when I sort of dove into that clinical lab, there's a part of me that felt like I was a little bit leaving science and research to become, more focused on the clinical application, the clinical side of things. And to some extent, I absolutely did that for a while. I was entirely focused on launching this clinical lab, developing novel genetic tests, running operations. We built the lab from me, N of one, up to a 50-person team in this clinical lab. And so for a number of years, I was very focused on just launching this lab and launching novel tests and the clinical care side. Now, over time, as you run a clinical lab, all those samples are coming into you. You're doing genetic testing. You're identifying novel causes of disease in terms of new genetic variants and determining which are pathogenic and which aren't, determining which phenotypes have underlying disease causality and which don't. And it really became a ripe foundation for writing papers on the work that we were doing. And so I started, being able to publish on our work in the clinical lab, being part of research studies that were using the results of our work. And then over time, NIH became more interested in genomic medicine as a primary field of research. And so I started working with colleagues, Robert Green, others, where we had studies to look at introducing genomics into the practice of medicine through studies like MedSeq and BabySeq and other programs. And so I started to, in some ways, get back into the research realm, having spent a bit of time launching this clinical lab, but then really using it as a platform to study the introduction of genomics into the practice of medicine. But along the way, I also realized the sheer inefficiency of some of the things that we were doing. We would identify novel variants or variants that we had to search the literature and find bits of evidence and information. And it was extraordinarily labor intensive to find information on variants that might be in some embedded paper somewhere. And the amount of work we were doing, and as the technologies began to scale and we converted from Sanger sequencing to next generation sequencing, now we are generating lots of data, lots of variation, and the ability to manually interpret these variants became a huge bottleneck. And that was when I started to think about, all right, there's got to be an easier way to do this. And instead of all these clinical labs operating in silos, what if we worked together? What if we shared our own efforts in researching these variants that we needed to report back to patients? And that's when I started to communicate with other clinical labs and start to think about shared approaches for how we could share the work we were doing. We're also realizing that different labs were interpreting variants differently. And so patients were not getting accurate results. And so by coming together as a community, we could not only reduce the labor involved in the work we were doing, but we also could improve the accuracy of the work we were doing. And that's when I sought out applying for, my own grant funding to build, a sort of consortium of laboratories that would share data and classify variants with rigorous standards. And that ultimately, developed into the ClinGen program that we now run today.
What was your experience kind of launching ClinGen, and what kind of challenges were there, like kind of at the beginning, and what are the challenges that kind of still exist today as well?
Right. Yeah, in the beginning, the question of whether all of these laboratories were truly willing to voluntarily share their data I remember talking to many companies that had all sorts of interesting financial models for how to get laboratories to share their knowledge and their variant interpretations. And there were all these complex models. Well, for every variant you share, every time anyone uses that variant and its interpretation, you will get paid a small amount of money, and there'd be this whole sort of financial network. And I just said, it's never going to work. No one will ever share data if they can't get money for it, and that just fundamentally isn't how this field should work. There should be a pre-competitive space where we can share the knowledge base, the evidence, and then laboratories can make profits on services on top of the shared knowledge base. And so I, sort of abandoned these financial models that people were convincing me was the way to go, and went down the route that we can convince them to voluntarily share this data. But it was a big risk. We weren't sure that was going to work. And we tried a couple different approaches, because the question was, where was this data going to go? And, in the beginning, we partnered with a colleague who had a involvement with a company that was going to make a database that anyone who donated to the database would have free access, but others would have to pay for it. It was still a little bit of a mixed model, and I was concerned. And then, a group of us approached NCBI at NIH about their willingness to develop a database to house this information. And NCBI, it had at that point in time been a really trusted resource and institute for creating highly valuable genomic resources for our field and sustaining them. So it was, in my mind, a much better model of something that wasn't tied to commercial entities and had a reputation for sustaining, things that were useful to the community that they had built. And so we thought that that was a much better model. And we began working with NCBI on what ultimately became the ClinVar database. And they built this database with the input of all of these clinical labs, coming together saying we were willing to share our data in that database. And I also, the first grant that we wrote included funding for about 7 or 8 clinical laboratories to really help provide the bioinformatics resources and covering some of the staff that could spend time really figuring out how to do this. What was the fields that we needed to share and how to get that data out of their systems and submit it and be sort of the guinea pigs of this process. So we actually funded, not a lot of money, but enough to justify some time and effort in helping build this platform. In the second round of funding, we ultimately didn't fund anyone for the actual sharing of data. It became part of their operational expenses. And that is how it works still to this day. And the funding that we have now really funds the expert panels that can resolve differences between laboratories and really apply a sort of another level of expertise to the process.
You mentioned like data sharing, and I think over the kind of the past year, we've kind of seen that kind of accelerated in terms of the pandemic. Do you think that we're kind of on the right path, and kind of what are the challenges that exist now that we kind of need to resolve in terms of data sharing?
Yeah, I think that the major challenges in data sharing today are several-fold. One, the most valuable data that we need to share is really individual level data from patients, from research subjects, from individuals, where we can get both genomic and other omic data and their health information and correlate it. But when you're getting patient or individual level data, the privacy and security of that data becomes a big factor. And having consent and permission to share, not wanting to compromise the identifiability of those individuals and risk loss of their privacy. And so there's a constant tension of really helping these individuals share their data to advance medicine, to advance our understanding of human health and disease, yet also do our best to ensure that their privacy is withheld or upheld, and that we're not compromising anyone's willingness and what they want to share. So that's one major challenge that we have to overcome. And I think transparent engagement and consent of individuals is how we can best tackle that, as well as ensuring we have platforms that we can protect this data appropriately, yet still share it robustly, which is also a challenge. The other, critical aspects are really thinking about the standards. How do we collect this data? What are the fields? What are the standards for annotating it? Because if we really want to learn from it, we have to bring data from every corner of the globe. But if everybody's collecting it with different standards and file formats and methods of access, then it becomes very difficult logistically to actually share that data and make use of it. And so that is one of the reasons that I've spent a fair bit of time over the last few years involved in the Global Alliance for Genomics and Health, really working with that organization to instantiate standards and policy frameworks that are applicable across the globe, and that really engage major driver projects and other efforts across the globe, so we can get everybody working in the same sort of dialogue and language and standards, so we can, do this as a global community.
You were involved in the ACMG, like AMP kind of guidelines for interpreting variants, and I think that's something that I extensively studied when I did my master's. I feel like I know it back to front.
Yeah.
Are there kind of any plans to kind of update this these guidelines?
Yes, we are actually in the process of updating those guidelines. So a new committee was convened with a number of members from the original guideline that we published back in 2015. It's co-chaired by Les Biesecker and Stephen Harrison. I'm involved, and so are other, a number of other individuals. And we've been working on this for actually over a year now. So we're hoping towards the end of this year or early next year to come out with the next version of that guideline. It will be more quantitative. It will be a point-based system that's aligned with a Bayesian framework. And so really working to continually move variant classification to a more objective and quantifiable framework from and less expert opinion and subjective. So that's the shift we're making, and we continue to refine and evolve the rules for variant classification to make them as objective as possible.
What has it been like this time around compared to kind of like when you initially released kind of the guidelines? What has kind of the process been like this time?
Yeah, I think the process has been really thinking hard about how can we use evidence to guide the rules. So instead of it being, well, what do we think is strong evidence? Oh, segregation in families is strong evidence. We'll give that strong, or supporting or moderate, but instead to actually use the statistical framework to say, well, what is the statistical likelihood that a variant in that location of the genome is actually, causal for this disease? And a LOD score is a statistical framework. And so we can use certain types of evidence that are more quantifiable to then code them with scores that are based on their actual statistical evidence. Same would be true of a functional assay where you validate it against known variants that are either pathogenic or benign, and based on the accuracy of those functional assays against, variants that have been classified without functional assay with other genetic evidence, for example, we can validate those assays, and then based on their performance in a quantitative way, apply a Bayesian point-based scoring system to that evidence. So, as we dissect these different types of evidence, instead of using expert opinion, we can really use the data itself to drive, how much evidence each of those pieces of data is worth.
Yeah. I mean, well, I look forward to reading it when it comes out. If we just kind of go back to your career, and I've had to write this down because it's a long list. Now let me read it out. You are currently the medical director of the Clinical Research Sequencing Platform at the Broad Institute, as well as the Chief Genomics Officer at Massachusetts General Hospital, and also you're a Professor of Pathology at Harvard. How do you do it? How do you do all of that?
Well, on the one hand, I do wear a lot of hats, and I'm involved in a lot of different projects and programs. On the other hand, they all coalesce around a common theme. So, my research is really around building building resources to support the community in the application of genomic medicine. And so my involvement in ClinGen, in Nomad, in our rare disease center that Anna Luria and I co-lead, where we're working to discover novel causes of genetic diseases. These are all, projects that contribute to improving clinical genomics. And then my role as medical director in the Broad's clinical lab is ensuring that we have a way to take the high throughput genomic sequencing, performed at the Broad and allow anyone access to that technology at a scalable level and at low cost. And then my role in Mass General Hospital as the Chief Genomics Officer is really about delivering genomics to patients. And so in that role, working with the clinicians to understand the barriers to ordering genetic tests for their patients. I ran the Mass General Brigham Laboratory for Molecular Medicine for 17 years before I took on this Chief Genomics Officer role. And in that laboratory environment, we, developed and launched every test there was, from, single gene sequencing on up to whole genome sequencing. Yet still there were so many patients not getting testing for various reasons, no genetic counseling, physicians not knowing how to order genetic tests or not knowing how to interpret the results, insurance, not paying for it, various barriers. And so my decision to take on this Chief Genomics Officer role was really about working on the front lines to improve the ability for patients to get these tests. And so we've, really working at that end. So yes, there's a lot of different things that I do, but they all come back to a central theme in, providing genomic medicine at the end of the day, and everything, every tool and resource we need to get there.
How do you feel like your kind of like previous kind of career and experience kind of has like kind of put you in this position essentially? And do you feel like you're kind of in your kind of happy place as well? Like this is like the perfect job, but like, yeah. Do you feel like it suits you like really well?
Yeah, I mean, I think what I found the most satisfying from the early stages of my career was recognizing that you can accomplish so much more working as a team. And so very early on, being very thoughtful about who I recruited into my lab, and making sure they were talented and capable and driven, but then enabling them to be independent and not, I'm not a micromanager. I bring talented people in, make sure they have basic guidance, but then allow them to really flourish and develop their own skill set and capabilities. And that continues throughout my career today. The only reason I can get done, half the things that I do is because I have just an amazing team of people that work with me, that each and every one of them are entirely capable, skilled, and very productive. And it is the global team, across both global organizations like GA4GH, also local teams that are doing that work. And we all work together. And it's really a teamwork environment. And it is that teamwork that are, the most critical things. The things I enjoy about my job is being to interact with all these just amazing people that do the work of the whole organization. And that brings me the most satisfaction in my career.
In your experience, what has it been like being like a female in such prominent roles? And like, have you experienced kind of any challenges, particularly in your early career and now, in that regard?
I mean, overall in my career, I haven't experienced significant challenge in being a woman. There was probably one position that I ultimately did achieve, but wasn't put in that role for a number of years after I probably should have been. And so, there was maybe one career step in the ladder that might have been affected by that. But overall, I work with people that respect the work that I do, and are willing to, recognize that work. And so I think I think the two things that I focus on are, finding those people that are team players and will respect you for your contributions. And it doesn't matter what your gender or your race or your background is, but really respects you for your contributions, and aligning myself with those individuals, and also adhering to those same standards myself and how I think about my team and the work that they do. And the other thing I have always done is gone towards the things that I enjoy day to day. When I mentor younger individuals, I say to them, there are two key things that you want to do. One, go into a field or a career that when you're at a cocktail party and someone asks you what you do, that you feel proud of whatever it is, that you can talk about it with excitement and feel like you're contributing to society, or to, and that you feel good about what you say you do. But then equally important is that you're in a job where the things you actually do all day long when you show up at work, you enjoy. For myself, I'm in meetings and phone calls all day long. It's like 40 hours of Zoom and meetings, particularly in the pandemic. It's literally all on Zoom. But I'm a people person. I enjoy that. I enjoy the teamwork. I enjoy problem solving as a group, interacting. That's what drives me. And so day to day, that's what I enjoy doing. Is, whereas someone else who's not a people person in my particular role would not be happy, even if they thought that it was an important job to be done. So I think those are two things: is finding something that you enjoy doing day to day in terms of what you're actually spending your time doing, but also feeling good about it. And then, the advancement will happen because you enjoy your job and you do it well, and then you'll be productive and you'll, flourish.
Yeah.
Does that make sense?
Yeah, no, that cocktail party thing is good advice. You're also involved in the All of Us research program. Would you be able to just discuss that a little bit and kind of the purpose of that program?
Sure. Yeah, the goal of the program is really to recruit individuals to be part of a large cohort where we can gather information both from medical records and personal, surveys to those individuals, as much information we can about those individuals, and also generate data, genomic sequencing, other, metabolomic, proteomic. Over time, there'll be lots of data generated on these individuals, as well as information collected, and then be able to use that as a rich resource of learning about human health and disease that can, continue to power our understanding of genetic diseases, and not just genetic diseases, but environmental influences on health and disease, other factors. So that's the overall premise. A key goal was really making sure that this cohort was incredibly diverse, even more diverse than our own US population. So a lot of emphasis has been on recruiting individuals who are underrepresented in biomedical research. And that's not just race and ethnicity as factors of diversity, but also age and demographics and socioeconomic status, gender, all different factors that allow us to have an incredibly diverse cohort that we feel will be most important to learn about information that will become applicable to all individuals from any background. Now, part of that program and generating data on these individuals is to think about return of value. So these individuals are contributing their own data to this program. What are ways that we can return value back to them? And so since we're generating sequencing data, one decision was, well, let's return both traits of interest, ancestry, and some of the fun traits of that we can return, eye color and different things like that. But then also, if we find information that is actionable to those individuals and could improve their health by intervening, then we want to return that information as well. And so the 3 funded genome centers, and we are one of those genome centers at the Broad Institute in partnership with Color and the Mass General Brigham Laboratory for Molecular Medicine as a team, we're participating in generating the sequence data and also interpreting those results so that they can be returned back to participants when we find, clinically actionable information.
What are some of the kind of ethical issues associated with that? Because I think one of the main things that come to my mind, obviously, is that when you get your genetic data, you're not kind of just learning about yourself. You're obviously learning about like potentially things that could be in your family as well. So kind of what other kind of challenges are there, and how are they kind of being addressed?
Yeah, you're absolutely correct. When we think about genetic data, it is sometimes unique in that it has impacts for other family members. And so that is something that's a factor. At the same time, there are very few examples where if you find a genetic variant in one individual that you can say with 100% certainty that their family member also has it. Usually, it's 50% likelihood, for example, for a dominant trait. There's some exceptions related to mitochondrial variation and other things, but most of the time you cannot still say for certainty that a relative, even a first degree relative, has the variant. And so while on the one hand, their risk of having the variant would be much higher if that first individual has it, it's still not a guarantee, and it still allows some autonomy of those family members to decide what they want to do, and whether they want to learn that information as well. But at the same time, we have saved the lives of individuals by identifying, actionable information early on so that we can prevent the onset of serious disease. And so, on the one hand, you can say, well, maybe somebody doesn't want that information. But in our experience, in the course of offering this information to individuals in various both clinical and research contexts, most individuals do want to learn this information, would take action to improve their health. And so I think on the net, we certainly feel that the benefits outweigh the risks, but each person makes their own decision whether they want this information back or not. And then each family member of those individuals also gets to make their own decision if their family members share their personal information.
I think it's a really kind of exciting time for genomics, particularly with all the kind of research projects that are emerging and ongoing. Is there anything in the field that you're kind of excited about and kind of like interested in watching evolve?
Yeah, I mean, I right now for the last few years, we've been focused on knowledge sharing, ClinVar and all the labs sharing their interpreted variants. I'm now involved in the Nomad database and getting allele frequency data out to the general community. But I think what I'm excited about now, which has been a struggle over the years, is really getting these rich resources of case level data accessible in broader environments. And the All of Us research program is one example of that. But there's cohorts being launched all around the world that are recruiting individuals into biobanks and other research studies. UK Biobank is a great example. Their decision early on to make that resource accessible to the global research community has fueled so much discovery and understanding of human disease from that. Even now, we would consider it a small resource compared to some of the others being launched. And really, what we really need to think about is the global collection of these biobanks and cohorts. And so I'm most excited about the potential for what we can learn by a federated network of global cohorts around the world that will just fuel another level, exponential level of our understanding of human health and disease.
Yeah, no, it's definitely going to be exciting. If you kind of were to look back at your career, and obviously your career is still going, but so far, are there any kind of missteps in your career that you kind of learned from and that's kind of built your character?
Yeah, it's a great question. I don't know that there's any major missteps. Perhaps I would have been smarter about more computer training early on in my career. I I have a lot of software engineers and computational scientists in my team. And, it does challenge me because I don't have a really strong fundamental training in computational science and software engineering. And I have to, really heavily rely on the managers of those individuals to oversee those teams and to explain to me, different factors, so that I can put budgets together and understand what our cloud compute costs are going to be and what are the best technical solutions. So, maybe if I was to go all the way back to college instead of minoring in physiological psychology, I would have minored in computer science.
Yeah.
That's probably one of the only, missteps that I can think about right now in my career.
Are there any sort of kind of notable people throughout your career that have kind of inspired you and kind of helped you along the way?
Yeah, I mean, I wouldn't say there's, like one single person or even a small handful of people, but lots of people along the way, that I interact with, that are just good people, and really taught me that, doing things for financial gain doesn't necessarily bring you more happiness than doing things for the good of society. And, I think watching the excitement in my scientific mentors, my own parents and their incredibly strong work ethic that I learned from, as a child and continued throughout my career. I think there are people along the way that I observed, and I thought were role models for showing me just the joy that you can have from just building things that are useful for the community that you work within, and the joy you can have from just being part of teams that are doing really amazing stuff. And so I wouldn't say that there are, particular people that were that critically instrumental, but just, in general, my interactions with so many different people that showed me, the joys of what could be accomplished.
Yeah. I mean, outside of your career, like, what do you kind of like to do in your spare time? I can see like a board in the background with loads of bases on it. I'm like, you still like studying genetics?
Yes, I was teaching HGBS nomenclature to one of the software engineers in my team the other day on my whiteboard. But in terms of the personal side of me, so of course I have two amazing children who I spend as much time as I can as they will allow, watching them grow and develop. But beyond my own children and my husband, I also enjoy lots of stuff out of doors. I ran a mountain club in graduate school for a number of years, and we did backpacking and canoeing and mountain biking, wilderness first aid, lots of trips that we went on. And I still, to this day, we camp every year, sleeping in tents and using outhouses and so on. So I love the out of doors. I also play ultimate Frisbee. My husband and I both play ultimate Frisbee. So, and unfortunately the season got canceled due to the pandemic last year, but we're hoping this year to be able to get back in the groove of playing that. So it's, one of my sort of side hobbies, in addition to just in general, trying to spend as much time outside riding my bike or hiking in the woods, those sorts of things.
Who's better at ultimate Frisbee, you or your husband?
It was actually my husband who introduced me to the sport when we were in graduate school. Right after we got married, his team needed some extra women. It's largely a co-ed sport, and they needed women. I'd never played before, but I had experience in field sports, so I joined their team. It was called Sudden Death. And played with them, and they were some of the most amazing ultimate Frisbee players. They were part of the group that started the sport in the 70s, early on. So I learned from some of the best players, and then continued on. I actually even continued. My husband sort of left Frisbee playing for competitive cycling for a while I continued playing on my own on various teams in the Boston area as a club sport. And then eventually my husband came back after he sort of gave up competitive cycling for the most part and joined the team that I'm on today called Toads. So we're a club team in the Boston area, and I, continue to play with them, as does my husband.
I only recently realized how kind of competitive like Frisbee is.
Yes. Yeah, it's a huge sport and can be incredibly competitive. Actually, an interesting story was my boss at the Harvard Partners Center for Genetics and Genomics, Raju Kuchilapati. His son was an ultimate Frisbee player. And Raju and his son started the professional league in the US and ran that league for a while. And David, his son, I actually played Frisbee with on some of the pickup leagues in the winter when we were just keeping ourselves active over the winter season.
All these scientists seem to be really good at Frisbee. I better get practicing. This is a bit of a silly question to end on, but if you could turn your kind of life and career into an autobiography or a film, what would the title be? And I've thought of one during this as well.
Well, I'm good that you might have come up with one, because this is a hard question. Something silly like teamwork makes the dream work or something.
Like tagline. I was thinking of something along the lines of like many hats or something like that, because you obviously said that you wear many different hats, and I think that would be kind of cool.
Yeah, that would be a good one too. Many hats. I definitely wear a lot of hats throughout my both career and the different jobs I do today. So that's a good one. I like that one.
Thank you so much for joining me today, Heidi. It's been really interesting and also inspiring just to kind of listen to you, especially you and having a child and going back to work so early.
Yes, not the advice I give to my young female pregnant members of my team, come back after 5 days, but not the best work-life balance advice. But I learned from my mistakes along the way as well.
Thank you so much. It's been great.
Well, it's a pleasure to speak with you.
Hello everyone, if you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, broad-LMM-Genomics.com. I hope you enjoy.